My name is Jae. Yeah, some people say like Dr. Yoo, but please call Jae. And I'm really honored to be here presenting our research of uveal melanoma at this symposium as a junior scientist. It's really great opportunity to learn more this cancer. So thank you, the CURE OM community, and thank you, Sarah and Lauren, to organize this meeting. So as Sarah mentioned previously, I am recipient for CURE OM, the research award in 2016. And our research mainly focuses on understanding molecular mechanism to control like tumor growth and metastasis. Today I'm going to talk about the very specific protein. Name is the Arf6, which might be the potential molecular target for uveal melanoma. So before starting, I want to mention this one. As you can see me, I'm Asian. And actually, I was from South Korea, and English is not my first language. So during my talk, like if you have any question, please interrupt me and ask a question. Maybe your question would be great. So, okay, uveal melanoma. Everybody know this one very well. The most common primary ocular malignancy, about 50% patient will develop metastasis, usually to the liver. And unfortunately, there is no effective treatment for metastasis uveal melanoma right now. And one is interesting thing for us, this one, mutation. Actually, activating mutation in G-alpha-Q gene, GNAQ or GNA11 in over 90% of case uveal melanoma. I'm going to talk about more detail this one. With the genetic landscape such as chromosome alteration and gene expression or copy numbers characterized by a lot of the different investigators. As you saw yesterday, the Dr. Scott Woodman, his work is great, one of great work for identification like genetic landscape in uveal melanoma. First, I want to talk very little like basic concept in biology or cancer biology. First is a central dogma. It's DNA, RNA, protein. Everybody know DNA, RNA, protein. DNA stores genetic information in cell, and actually DNA make protein. And genetic landscape focus on DNA in RNA level. And our research focus on protein level. And the final goal is understanding this protein function at the molecular mechanism. So actually protein actually do something in the cell. And what we believe, if we have more understanding this protein in the cancer, that allow us to have more opportunity to develop drug. So our research mainly focuses on this protein level. And I'm going to talk about specific protein, name is Arf6. Okay. Another concept is oncogene and tumor suppressor in the cancer. So you can easily think about the oncogene. Oncogene is accelerator in the cancer. And tumor suppressor is brake in the curve. If you have activating oncogene, what's happen? Make a cancer from normal cell. If you lose the tumor suppressor in the curve, in the normal cell, what's happen? Finally, cancer. So this is a very complicated step to get the cancer from normal cell. Sometimes like few oncogene activation and tumor suppression loss is enough to get the cancer. Sometimes like a lot of different oncogene and tumor suppression you need to get the cancer from normal cell. The reason why I'm talking about this oncogene is because of GNAQ and GNA11, which I mentioned previously in the slide. The activating mutation in GNAQ and GNA11 over 90% to uveal melanoma. And GNAQ, GNA11 is oncogene. Let me talk more detail about GNAQ and GNA11. GNAQ is GQ alpha subunit. This picture is showing that one, and GTP is protein, and definitely oncogene. What is GTPase protein? And you can easily remember this one thing. GTPase protein has GDP bound form, two form actually. GDP bound form, which is off inactive form, and GTP bound form, which is on active form. If you have activating mutation in GNAQ or GNA11, what's happened? Always on and keep activating downstream signal pathway in the cell. So this one, you can remember like activating mutation in the gene, and finally this gene make constitutive active protein, GNAQ or GNA11. This is oncogenic GNAQ, GNA11. So, and constitutive active form of GNAQ, GNA11 make downstream signal pathway. Now question is, what is signal pathway? This is most our interesting to figure out, understanding what is pathway, what is molecular pathway. Look at this. The signal pathway or transduction in serial molecular event upon stimulation signaling. This is in the cell, and upon stimulation like signal and serial molecular event you can see here. And finally, cellular response you get. And some of cancer, probably most of cancers have like some of the signalings are amplified by oncogene or tumor suppressor, loss of tumor suppressor, or other like protein. So again, our research focus on this step. And this picture is showing very complicated picture. So actually this signal pathway in uveal melanoma, I believe there is more, but this is a well-known signal pathway. And very complicated pathway, but I want to point out this one. As I mentioned, G-alpha-Q, activating mutation, always activation. And what's happened? Chain reaction, a lot of different signal pathway activated by this protein. But you can remember just a few pathway, PKC pathway, MAP kinase pathway, pathway. These pathways are very, very important for cancer cell, especially uveal melanoma cancer cell growth and proliferation. And now a lot of investigators are trying to target different signal pathway. But as you can see here, what is the best way to target in this pathway? Probably this one, right? But unfortunately, my understanding, there is no drug or inhibitor to target this oncogene directly. So our goal is, let's study more about this one, and then we will see what's happened. Still, you can see a lot of error in here, but we don't know what's molecular mechanism, how this oncogene activates a whole bunch of different signal pathway which are important for cell growth and proliferation. So this is our main goal, understand this like molecular mechanism. And this is side story. Whenever I'm showing this kind of a picture to my kid, and my kid told me like, hey, Daddy, are you trying to understand this arrow? Or he's like, that is boring. I can draw this arrow for you, but that is not true. Even though just one arrow, but there are a lot of molecular mechanism in there. Just a simple arrow, but we don't know how to work. We can make a draw, but still there are a lot of complicated molecular mechanism in there. And let me switch it to a little bit like a topic to Arf6, which I'm going to talk about. This is same GTPase protein like GNAQ and GNA11. So Arf6 has kind of two form, GDP bound form, off, inactive, GTP bound form, which is active form, on. And this Arf6 GTP make signal transduction like GNAQ and GNA11. Our lab has been studying about Arf6 in different cancer and different disease. And this picture is just showing, actually this is cell, and a lot of upstream signaling can activate Arf6. It depends on cancer cell and cell type. And eventually this active Arf6, which is Arf6 GTP form, usually regulates cell motility and invasion of cancer cell and cell proliferation. I'm going to talk about more detailed invasion, but this is Arf6 general function. There are a lot of signal can activate Arf6, which is Arf6 GTP form, and this active Arf6 controls the motility and invasion and proliferation depends on cancer cell. Why we start to study Arf6 in uveal melanoma? Just wondering? No. We have a background start to study Arf6 in uveal melanoma. Let me show this one. Probably you know this one. It's a gene expression profile. And this is one of the prognostic factor in uveal melanoma, class 1 and class 2. Class 2 is bad prognosis, the type, the gene expression, and this table is showing gene list in class 2 uveal melanoma. And unregulated mean is more RNA, possibly more protein. And downregulate mean is less RNA, possibly less protein. I want to point out this one. ROBO1 is one of gene in here. Please remember ROBO1. What is ROBO1? Just receptor. And few years ago, what we found for Arf6 function in cutaneous melanoma, we found active Arf6, which is GTP-bound form, promote invasion and metastasis of cutaneous melanoma. And ROBO1 here. ROBO1 make inactive Arf6, which is GDP-bound form. And actually that one can inhibit invasion and metastasis. This is rationale why we start to study of Arf6. First, it has been demonstrated that G-alpha-Q signaling activate Arf6, which is Arf6 GTP-bound form. That is one rationale. The second rationale is ROBO1 loss. The loss of ROBO1 expression is one of the marker to predict progression to metastasis. Wow, this is background. I mean, my mentor told me like, Jay, that is 2013 when I was a graduate student, and he said, Jay, there is a smell. So I don't know what smell are you talking about, and he told me like, we need study Arf6 in uveal melanoma based on this background, and then I start to study this Arf6 specific protein in uveal melanoma. And first, what we have to make sure is protein, because our research focus on protein level, so make sure protein is there. Arf6 protein is there. Sorry, yeah. And we check Arf6 protein. This is not gene expression and not gene, just protein. Make sure Arf6 protein is there before starting study Arf6 in uveal melanoma. And we got this sample, patient sample from Bill Harvard, and this each individual lane indicate the patient and non-tumor here, and about 10 human melanoma sample we got from Bill, and we check Arf6 protein. You can easily see band in here, and then intensity of band dictate how much protein of Arf6 in here. So you can easily see compare, so non-tumor is less intensity. However, you can see high intensity of like a band, which is Arf6 protein over uveal melanoma tissue. So this is a quantification. This each dot indicates like each one sample. So 2 sample, 10 sample we saw. Our question was, Arf6 protein is there? Yes, it is. And what we found, even Arf6 protein levels are increasing human melanoma. So wow, now time to start a study for Arf6 in uveal melanoma. And our basic question is here. why they need a high Arf6 protein in the uveal melanoma, and what is function? If Arf6 has very important function for uveal melanoma, next question probably, can we target Arf6 in uveal melanoma? So first, make sure what is the role, what is the function of Arf6 in uveal melanoma. As I mentioned, it's a lot of band, but I can explain that. So, as I mentioned previously, Arf6 is GTPase protein, two form. GDP bound form, which is inactive form, and GTP bound form, which is active form. Even though human melanoma sample we saw high Arf6 protein, we want to make sure that Arf6 is active, which is GTP bound form. We got cell, the human uveal melanoma cell. This cell actually isolated from human melanoma sample, primary tumor. And we got cell from Scott Woodman group. Actually, it's two cell line, but I want to point out just one cell line first. This band, first this one. This is normal just uveal melanoma cell. And this band dictate GNAQ protein. You can see here. It's good protein expression and good protein in here. This cell has GNAQ mutation, activating mutation, and we were reducing this GNAQ protein. So you can see, actually no band there, which means we got rid of that protein, GNAQ. And then go up. We check Arf6 GTP level, which is active form. This is normal uveal melanoma. Arf6 GTP level is good. When we reduce the GNAQ protein, what's happened? Goes up. Arf6 GTP levels go down. That is interesting. Also, we use different reagent to reduce the GNAQ. Same thing. When we reduce the GNAQ protein, Arf6 GTP, which is active form, goes down. Okay. And we did, this is one cell line and the other cell line we did is the same, exactly same pattern we saw, different cell. And what we did, we did opposite experiment. We took normal cell. This cell has no mutation of GNAQ. No mutation, just wild type. No mutation. What we did, just the normal situation, this normal cell, Arf6 GTP is there. Again, this band dictate amount of Arf6 GTP here. We put just normal GNAQ protein in here. Well, it's okay, same. However, when we put oncogenic GNAQ, which found in uveal melanoma, into normal cell, okay, that's what's happened. Arf6 GTP levels go up. Arf6 is activated by oncogenic GNAQ, which is found in uveal melanoma. So now we have two. What is two? High protein level in uveal melanoma. Second thing is Arf6 is activated by oncogenic GNAQ. So now, what is the function for Arf6 in this specific cancer cell? Okay, first we check cell proliferation, this specific cancer cell. Now please understand, now we use cancer cell. Cancer cell first, and then we check cell proliferation. Because one of the characters for cancer cell, uncontrolled cell proliferation and cell growth, we check first that one in terms of Arf6 functions. We use a lot of different uveal melanoma cells which have GNAQ and GNA11 mutation. And this is one of the cell and two cell line. We can go here. This is individual cell. It's the same cell, but we did manipulation individually. You can see those control uveal melanoma cell, and we trying to reduce the Arf6 protein to check Arf6 function in the cancer cell. Also, we use different reagent to reduce the Arf6 protein. Just focus on these three, right? Normal and Arf6 reducing cell 1, 2. And then we were plating them, this cell, in the 2D culture and 3D culture system to check cell proliferation or cell growth. 2D culture is just plating the cancer cell on top of the plastic tissue as a monolayer and then check cell proliferation. That is 2D culture system. You can see here, normal just uveal melanoma cell growing very well in 2D culture. However, when we reduce the Arf6 protein 1 and 2, you can see proliferation goes down in this specific cancer cell. And we use GNAQ as a positive control. What is positive control is, as I mentioned previously, GNAQ oncogene to make a lot of different signal pathways which are very, very important for cell growth, cancer cell growth and cancer cell proliferation. And if we get rid of GNAQ, definitely all signal pathway will be inhibited by reducing GNAQ, and that signal pathway is so important for cell proliferation. So that's why we use GNAQ as a positive control. But this is quite surprising. You can see here Arf6, when we reduce the Arf6, proliferation goes down. You can compare this normal uveal melanoma cell in terms of proliferation. And everybody knows cancer cell growing in the body 3D structure. It's not 2D at all. It's not monolayer. So what we did, we mixed with matrix with cancer cell. And then this is normal cancer uveal melanoma cell, and then we monitor cell growth. And you can see here is good tumor cell growth, we can see. However, when we reduce the Arf6, look at that, it's not much good cancer cell growth in here. Also, it's the same like Arf6 reducing cell, but we use different reagent, also GNAQ as a positive control. So based on this experiment, our conclusion is Arf6 is so important for uveal melanoma cell proliferation. So that's why uveal melanomas have a high protein Arf6 and active Arf6. Next question is, what is molecular signal pathway? What is molecular levels mechanism? I keep showing like a lot of arrow and cartoon, but I cannot show whole like individual data. But bottom line is, Arf6, very interestingly, Arf6 control LC-PKC signal pathway, MEP kinase signal pathway, YAP pathway, and even unknown pathway. These pathways are activated by oncogenic GNAQ in uveal melanoma, and this pathway is so important for cancer cell proliferation and cell growth. Eventually, Arf6 control all signal pathway. This is great. As I mentioned previously, a lot of investigators trying to individual signal arm to target to inhibit cell growth and tumor genesis. Now what we know, only one Arf6 control, like all signal pathway activated by GNAQ and GRPQ in uveal melanoma. So, wow, this is amazing. But still we have a question as a basic scientist. Still we don't know how. How Arf6 control all signal pathway, even though that is really good molecular target in uveal melanoma. End of experiment, what we found? This one. Location of oncogenic GNAQ in the cell. This is normal uveal melanoma cell, and I was making like very simple picture, uveal melanoma cell, and we check location of GNAQ protein in the cell, in the cancer cell. And you can see here like green indicate like GNAQ protein in uveal melanoma. When we reduced Arf6 protein, what's happened? Look at that. All GNAQ protein goes back at the cell surface. Now, makes sense. Because GNAQ, this oncogenic GNAQ, activating mutation GNAQ, need Arf6 to go appropriate place in the cell to make all signal pathway, which is very important for cell growth and cell proliferation. When we reduce the Arf6, GNAQ protein cannot go appropriate place in the cell. So that's why all signaling goes down and the cancer cell proliferation goes down. We saw. So this is, wow, this is is amazing. Like Arf6 protein is really amazing. And now I'm showing like just the cancer cell. But this cancer cell actually make a cancer in the body. So make sure if we target, if we reduce the Arf6, actually affect like tumor growth. And what we use, mouse model. And we took human melanoma, uveal melanoma cell, cancer cell, and we injected that cancer cell into mouse eye here, and then let them grow 5 weeks. And then we saw tumor in here, good tumor. Other experiment what we did, we were reducing Arf6 protein, which we did previous like cell experiment, and we injected that cell into mouse eye and let them grow 5 weeks. And then we saw actually no tumor. And then we quantify like a tumor incidency. Actually, there's normal cell, uveal melanoma cell, injected mice, had 12, 11 mice, had good tumor. Look at that. But when we reduced Arf6 protein, only 3 mice had tumor, and 8 mice, no tumor at all. And also we check the tumor size. Just the normal cell injected mice had a good tumor size, tumor growth, and then when we reduce the Arf6 protein, it's not, even though 3 mice, it's not big tumor at all. So based on experiment, wow, this is tumor growth I'm talking about in terms of Arf6. So based on this experiment, Arf6 is important for what? Tumor establishment based on that, and tumor growth. Now, kind of we got answer for that. What is the function of Arf6? And next question is, can we target Arf6? And this picture is showing the molecular structure. Arf6 protein is there, and this the specific like drug in here. So we want to make sure this inhibitor is actually working. First cell biology experiment, we treated this inhibitor and make sure this inhibitor inhibit Arf6, which is Arf6 GTP form. That this uveal melanoma cell is normal cell, good Arf6. Again, this band dictate amount of Arf6 GTP. Look at that. It's good Arf6 GTP level there. When we treat this drug, less Arf6 GTP here, which means good inhibitor for inhibition Arf6 activity. And then we check cell growth in 3D culture, just control normal uveal melanoma cell, and we treat this compound, and this compound is really good, working in terms of inhibiting cell growth in the 3D culture, and this is GNAQ location in the cell. So this pattern, what we saw this is exactly what we saw when we reduced the Arf6. When we reduce Arf6, what's happened? Tumor growth goes down, and not tumor growth, so cell growth goes down, and location, something like that we saw. So cell biology is kind of same pattern we saw, like when we reduced the Arf6. So what we need, next experiment is mouse work. So we injected like a cancer normal uveal melanoma cancer cell eye, and we divide by 2 groups. And 1 group just vehicle, just control, let them grow, 5 weeks. And 1 group, we injected cancer cell, and then we gave this drug through IP, interpenetrant injection, 5 weeks. And then what's happened? Tumor grows here very well. However, when we treat this drug, not good for tumor growth here, right? And then we check like tumor incidency. 9 mice had very good tumor. It's just control like normal mice, the cancer cell injected mice. But only 0.5%, 50% mice had tumor when we treat this drug. And we check tumor size. This is good tumor size, but when we treat this compound, what's happened? Tumor size is very small compared to control. So this is kind of like summary. Arf6 is very important for GNAQ, this the oncogenic G-alpha-Q location in the cell, and this complex actually make all signal pathway, which are important to cell proliferation and tumorigenesis. Now we have specific Arf6 drug, and this drug inhibit cancer cell proliferation and tumorigenesis. So I want to go back like Arf6 introduction. Arf6 is actually known to control cell invasion and cancer cell invasion and migration rather than cell proliferation. In the breast cancer cell and cutaneous melanoma cell, usually Arf6 controlling this kind of invasion and cell migration, kind of cell moving. What is invasion? This picture is showing like very complicated step to get the metastasis, and first the primary tumor there, and then to get metastasis, the angiogenesis should be there, and then invasion should be happen to get the metastasis, and then go to blood vessel, and then circulation, and then eventually go out from there and establishment and proliferation. So this invasion step is so important for getting metastasis. And again, Arf6 function is known to control this invasion. So our question was, so Arf6 also important for uveal melanoma cancer cell invasion and metastasis. What we know so far, Arf6 is important for cancer cell proliferation and tumorigenesis. Now another question is, uveal melanoma cell invasion as well. And what we did, we check invasion of this specific uveal melanoma cancer cell. We use exactly the same technique, like reducing protein using siRNA. And this normal cell, you can see like a green spot indicate invasive cell. And when we reduce the Arf6, what's happened? Less invasive cell, which means Arf6 is so important for the uveal melanoma cancer cell invasion as well. And this is quantification. Also, we treat like Arf6 inhibitor as well in this experiment. Exactly same pattern we saw like this. So now, okay, Arf6 is also important for uveal melanoma cancer cell invasion. What about metastasis? We want to see metastasis. So first experiment, like to monitor primary tumor, we use specific this technique. We inject the cancer cell into mouse eye directly and then let them grow 5 weeks. And we saw tumor cell here, and then we treat the drug, or like we use like Arf6 reducing cell for injection. Unfortunately, we couldn't see any metastasis when we used this technique. We injected thousand cells, we injected 100 mice, we couldn't see that. So we slightly changed our technique. We injected cancer cell behind our eye, which means retrovital injection. And 8 weeks, what we saw, this is the mouse imaging. Blue means cancer cell. You can see like a spreading of cancer cell in the whole body of like mouse. 8 weeks after injection, and we took out the liver from these mice. This black cancer we saw right now. So now we have a metastasis model. And now what we are doing right now as an ongoing project, we injected uveal melanoma cancer cell first, and then let them grow 3 weeks, establishment and grow well. And then we start to inject our Arf6 inhibitor. And this is now ongoing project. We are really wondering about what is result. And this is a summary. Okay, summary. So oncogenic GNAQ Arf6 is required cell proliferation, uveal melanoma cell proliferation. Also Arf6 is very important for uveal melanoma cancer cell invasion, and reducing Arf6 protein or inhibiting Arf6 with specific like drug reduce tumor establishment and growth. That is what we know so far. You need to take picture? Yeah. And this is the Dini. Dr. Dini was my mentor for my PhD and postdoc, and now he moved to Merck company right now. But still, he is doing like mentoring for us, and he's giving good feedback for our research. And Shannon is another my mentor, and I have good colleagues, the Ali Grossman and Wendy. 2013, when Dean was asking me to study uveal melanoma, at that times, I was the only person to study uveal melanoma in our lab. Now, I have team. They want to be Jedi to fight cancer. Yeah. Yeah. Also, thank you for my support, big support, my life and my research, my daughters. And we have a good collaborator like Navgen, the pharmaceutical company, and Bill and Scott and Crislin. Also like a funding support by CURE OM Junior Fellowship Award. Thank you, everybody, for attention, and I'm happy to take questions.
Maybe can I start with the question, and then I'll run around the room. Two questions for you. The Arf6 inhibitor that you're using in the lab,
yeah.
Is there an in vivo counterpart that's been used in humans? Is the first part of the question.
No, not yet.
Okay.
We still checking, testing the compound or other compound as well. First mouse, but yeah.
And then this question, the second part of the question then may be less relevant because of that answer. But yesterday we were hearing from Scott Woodman this idea of like whack-a-mole, like you hit the pathway over here, the other one comes up, and so the Arf6 target is nice because it's it's above those different pathways,
yes.
So you would
right.
circumvent the whack-a-mole. However, the other thing we know in humans is that that these pathways in normal cells are important. So you might want to inhibit them in a tumor cell, but do you have a sense, at least from the animals, when you're inhibiting Arf6, which is much higher up, what you're seeing, what some of the side effects are that you're seeing in normal cells?
That is a good question. I got a lot of question kind of that. Yeah, Arf6 is very important in normal cell as well. And actually what we did, we injected that drug. It's just normal mice. We couldn't talk with mice, how much they are sick, right? But what we check is just like pathology defect. But we couldn't see any pathology defect. It's just normal mice. When we injected, we actually injected 2 weeks, every day, and we couldn't see any pathology defect, which means probably okay, but we still keep testing that.
So 2 questions for you. One, when you got the metastatic effect that you wanted, and you injected at the 3-week point with the 2729, I think it was, were you injecting the primary tumor site or the metastatic site, or have you tried both? Does it matter?
That is a really good question. So actually, to get metastasis model, we injected different cancer cell. First, what we use a cancer cell from primary tumor. But it's really, we couldn't see any metastatic tumor at all. And then we switched the cell that is from metastatic cancer, human, and then we saw that way. So yeah, usually metastatic, the cancer uveal melanoma cell is better to see the mouse model, to see metastasis model.
Okay, and then my second question was for the ROBO1 marker, how much did that correlate with the greater than 90% GTP, GNAQ? So is the ROBO1 marker 100% correlation, or is that just one of the 12 and it's only 20% correlated, whatever it is?
That is a really good question. The region to start Arf6 because of ROBO1, right? And one of our project is what is ROBO1 pathway? That is our question. And then cutaneous melanoma, we know ROBO1 inhibit Arf6, but uveal melanoma doesn't inhibit Arf6 at all. And now we are doing some of study for ROBO1 project. Now we found a new signal pathway. by loss of ROBO1. Looks like uveal melanoma, ROBO1 doesn't control any Arf6, new pathway. Even though cutaneous melanoma, ROBO1 inhibit Arf6. Can you understand that? So that is kind of our ongoing project. Its bottom line is uveal melanoma, ROBO1 does not affect Arf6 GTP level.
Okay, thanks.
You worked exclusively in GNAQ. Do we know if any of this would apply in GNA11?
Good question. Yes, a lot of cancer cells which we use is GNAQ mutation. And some of the cell line we have GNA11. Now we are testing that one. And what I can tell you right now is kind of same pattern we saw. But still we need to test more cells which have GNA11 mutations.